Howdy, Earthlings! Today we are going to learn about the process called cell division, which is also known as mitosis. Here are the learning targets we will be focusing on for today. This includes a lot of new vocab, so hang tight, buckle up, but I will walk you through it step by step, so don't worry too much. This set of notes does integrate with drawing notes, which will help you better understand the relationships between all of these different vocabulary terms. Let's first start with the review of the cell cycle. We learned about the cell cycle and how it is a cycle of growth, DNA synthesis, which means reproduction or replication, and the process of cell division. There are 4 major stages of the cell cycle. This includes gap 1, synthesis, gap 2, and mitosis. Together, G1, S, and G2 are known as interphase, so interphase is followed by mitosis. Remember that in gap 1, this is where a cell is doing its normal required functions, it's growing in size, organelles are developing and multiplying, and this is where cells spend most of their life. There's a checkpoint at the end of gap 1 to make sure the DNA is intact and is not mutated, hopefully, and that there are enough resources to progress through the cell cycle. At the end of gap 1, or throughout gap 1, during gap 1, there is another phase that a cell might enter depending on its type, and this is called G0. This is a resting phase where cells perform the required functions, but they are not necessarily progressing through the cell cycle with the purpose of dividing. They are simply there to do their job. So depending on the cell type, you learn that G0 may be reversible. So a cell might be in G0 for a prolonged period of time, whether it's years or months. And then it also could be irreversible, where some types of cells, such as certain types of neurons, once they are born, they never go through the process of cell division. They are, once they die, they are permanently dead. All right. You learned about synthesis. Synthesis is a scientific word for to make new. And so this is where we are making new copies of DNA from the original set of DNA that's present. We will learn about this detailed process in one of the upcoming units. Again, synthesis means to make, so at the end of the synthesis phase, there are 2 identical copies of DNA present in the nucleus of the single cell. At the end of synthesis, there is a checkpoint where DNA is checked to make sure that it was copied correctly and hopefully there are no mutations. Gap 2 is another very short phase where there's more normal functions and additional growth, and it's final preparation for the process of cell division coming up in mitosis. So at the end of gap 2, there's another checkpoint to make sure, again, the DNA is correct and that there are enough organelles to divide it properly into 2 cells at the end of mitosis. And then we enter mitosis and cytokinesis to conclude. So mitosis occurs in 2 major parts. The process of mitosis overall is the division of the nucleus and the DNA inside of it. And the end process of cytokinesis is the final division of the cytoplasm and the cell membrane, which fully forms 2 complete cells. So the result at the end of the overall cell cycle is that one cell has divided to form hopefully 2 identical cells. With this review, let's now dive into, and also hopefully each cell has identical DNA. Now we're going to dive into the actual detailed steps of this process of cell division. If you take a look at this video, you can see that we start with one cell. The DNA, which is shown in bright red, is sorted and organized and arranged down the middle of the cell and then split apart so that eventually you have 2 cells. Each cell has 1 complete set of DNA from the original cell. But before we begin, let's start with some important vocab that's coming up. First, I want to help you recognize that there are 2 major classifications of cell types in your body. We will be learning about sex cells, which are sperm and egg cells. So depending on your biological sex, you might produce sperm or egg. And then your body cells basically are the cells that make up everything else, such as tissues, blood cells, neurons, etc. And mitosis, which we were learning about, is the process of making more body cells for 3 of the 4 R's. That includes repair, replace, and grow. So we need to repair broken down cells or damaged cells. We need to replace cells that are worn out and for the organism to grow bigger. And when we eventually learn about the process of making more sex cells, which is the process of reproduction, this is a different process that we aren't learning about today. We'll be learning about this in Unit 9. Further vocab, the cell that we start with at the beginning of the cell cycle and the beginning of cell division is called the parent cell. And the two identical cells that are produced by the process are called daughter cells. And last, some more vocab that's coming up. We need to identify and introduce some specific vocab related to DNA. We haven't learned a ton about DNA, and this is kind of one of those which comes first, the chicken or the egg kind of things. Do we talk about DNA first, or do we talk about the cell cycle first? But we need to do at least a little bit of pre-teaching about DNA vocab before we dive into the nitty-gritty details of mitosis. So we're going to do a short set of drawing notes to help introduce a couple of terms, including chromosome, chromatin, sister chromatids, and centromere. Throughout the different stages of the cell cycle and mitosis, there are different forms or structures of DNA. So we're going to talk a little bit about the DNA and what it's referred to as during the process of interphase, specifically during G1 and then G2, and we will also then introduce what DNA looks like and is described as when we enter the process of mitosis. So in your drawing notes, you can see we're looking at on the left side of your screen, we are looking at G1 first. This is the beginning of the cell cycle. So we have one cell, the nucleus is intact, all of the organelles are inside of that cell, they're just not pictured. But you can see that the nucleus is labeled, and the nucleus is where all of the DNA is located. So reading through the text, it says a chromosome. So prior to S phase, a chromosome consists of one long strand of DNA. You've learned about how DNA is a double helix structure and how it contains all of your genetic information. And if we were to illustrate what this strand of DNA looks like in a cell at the beginning of G1, it is very loose and uncoiled. And it kind of looks like spaghetti in a noodle, or spaghetti noodles in a bowl. So you've got long strands of DNA. And in a human cell, in your cells of your body, there are 46 of these. So I illustrated one there, but there are 46 total chromosomes in a human body cell. For the simplicity of this diagram, we're just going to draw two. Each chromosome is a different length. So for example, I drew one really long one and I drew one really short one. So these are two chromosomes shown at the beginning of G1. And because they are loose and uncoiled, they are what we call chromatin. During interphase, DNA is very loosely uncoiled like spaghetti noodles in a bowl. DNA at this stage is referred to as chromatin. So chromatin is a word that refers to DNA during the part of interphase where it is loose and uncoiled. And because of the fact that it's so loose and uncoiled and it's extremely long, and you've got 46 of these strands of chromosomes packed into that nucleus, it is impossible to distinguish individual chromosomes from each other using a microscope at this stage of the cell cycle. Then the cell progresses into S phase, which is when DNA is synthesized, meaning copied. So reading through the text associated with G2 right here, DNA is still in the form of loosely wrapped chromatin. However, each strand of DNA has now been replicated or copied. So let's write the word replicated. We'll be learning about the process of DNA replication next unit, Unit 7. The two identical copies of DNA are attached together at a structure called the centromere. Centro referring to middle, meaning it's the middle place where two copies are connected. Centromere. So remember that we have one really long chromosome, and it has been replicated or copied. And to help the cell keep track of all of this DNA and keep everything organized and sorted, the two identical copies are attached together at a structure called the centromere. And I represented that structure with like that big circle, that big ball. So that is essentially what we call the centromere. So I'm going to label that. You should be illustrating this in your set of notes, by the way, if you're not already doing that. So the centromere is a structure that holds the two copies of a chromosome together. And each of these two copies, now because of that there's two copies of them, we need to call them something else. We can't just refer to them each as a chromosome because there's two copies of each of that chromosome. So each copy within the replicated chromosome is called a sister chromatid. I do not know why the vocab uses, for example, we talked about parent cell and daughter cell, and now we're talking about chromosome and then sister chromatids. I don't know why sister and daughter were used in these contexts. It's just the way it is. So each copy within the replicated chromosome is called a sister chromatid. So I'm going to use a different color here to kind of emphasize that. So each of these strands of identical DNA are now called sister chromatids. Please don't get chromatids confused with chromatin. The sister chromatids are copies. Because they're loose and uncoiled, they are in the form of chromatin, loose, uncoiled DNA. You can also replicate your small chromosome. So there's my small chromosome. So you can see we have 2 chromosomes. 1, 2. This was 1, 2. We haven't changed the number of chromosomes. We've just changed how many copies of each of those chromosomes are present in this cell. There are 2 copies of chromosome 1 and there are 2 copies of chromosome 2, and the word to refer to each of those copies is called sister chromatids. Now as we enter the process of mitosis, which is the actual process of cell division, the DNA becomes tightly condensed or supercoiled when the DNA tightly coils up. So imagine you are wrapping a spaghetti noodle around a fork so that it's easier to stick into your mouth and eat. Or like imagine you had a really long piece of yarn or string and you wrapped it around your finger to easily organize it. That is essentially what's happening in this process of mitosis. So because this DNA becomes tightly condensed and wound up, it's like this. It's smaller in size now, but it's more visible too. So the DNA tightly coils up. Here's the small one. You can see the individual copies. So when you're looking at this under a microscope, you can actually see them as X shapes. Here is, again, I'm going to show you the sister chromatid. So we've got, here's one sister chromatid. Here's a second sister chromatid. And they were attached together at the centromere in the middle. Okay, we still have, here's one chromosome, here's the second chromosome. It's just that there are multiple copies of each of those. What is the purpose of DNA becoming condensed or supercoiled? It helps the cell keep the DNA organized. It helps, just like when you are wrapping up, if you are, for example, a knitter and you have lots of different types of yarn, you want to keep that yarn organized in yarn balls so that it's easier to organize and move things around and choose the yarn you want to work with. Same thing with DNA. So this is all of the vocab associated with DNA. Remember that chromatin is loosely uncoiled DNA. Chromatids are copies of each chromosome. And then centromere is the structure where the copies are held together. So those are the 3 major terms we talked about in this set of notes. All right, now let's dive into the process of cell division, which is called the process of mitosis. This will be another set of drawing notes, so watch the video and follow along carefully as you draw along with me. Let's illustrate the process of cell division. We are going to start at the beginning of the cell cycle in interphase. So that means we're starting in G1. During G1, the chromosomes are loosely uncoiled and they are not individually visible, which means that the DNA is in a stage called chromatin. When we were talking about DNA vocab, we mentioned the fact that DNA in a human body cell, like in the cells that make up your skin cells, for example, there are 46 individual chromosomes. We are not going to be drawing 46 chromosomes. We're going to keep it a little bit more simple. We're going to draw a cell with 4 chromosomes, and I'm going to use 2 different colors. So go ahead and draw this with me. I'm going to draw a really long red chromosome, loosely uncoiled in the form of chromatin, and I'm going to draw a short red chromosome. I'm also going to draw a long blue chromosome and a short blue chromosome. Let's go ahead and label our nucleus as well, just to make sure that we're following along with our understanding of where this is happening inside of a cell. Keep in mind that all of the other organelles are still present in the cell. We just aren't drawing them to keep it pretty simple. So we have 4 chromosomes in this picture. Let's go ahead and label that at the bottom of our cell, 4 chromosomes. And remember that in a human body cell, there would be 46. let's dive into the next phase. So at the end of S phase, remember that S means synthesis or to make more. During S phase, the DNA has been copied. So each chromosome has been copied or replicated. Replicated is the more scientific term, so we're going to practice using that word more often. Each chromosome has been replicated. The two identical copies of each chromosome are now called, and we used green for this in our last picture, they are called sister chromatids. And they are attached to each other via a structure called the centromere. Okay, so let's go ahead and draw our chromosomes. They are identical to the ones in the first cell we drew, except that there are now copies of each. So here's my long red one, and here's its identical copy, and they are attached at the centromere. You can use orange for that centromere if you want, like I used the color orange to write the word. Let's draw our short red and its copy with the centromere, attaching the two copies, the two sister chromatids. We have a long blue chromosome, and here's its identical copy attached at the centromere, and the short blue. So now you can see, again, we have 4 chromosomes in the cell. Just to make this easier for later, I'm going to use my green to identify the sister chromatids. So 2 sister chromatids basically represents 1 replicated chromosome. Now that we've reached the end of G2, we are going to start the process of cell division. Therefore, we are going to start with the first phase of mitosis. The first phase of mitosis is called prophase. During prophase, the chromatin structure of the DNA begins to condense and tightly coil to form the individual visible chromosomes. These can be seen under a microscope. When we draw our DNA in the condensed form, we need to remember that it's tightly coiled. So if we were to look at our long red chromosome, that means it's now looks a little bit smaller than it originally would have. You can again see that X shape, that traditional X shape that we think of sometimes when we look at microscope images and think of chromosomes. So that's the long red one. The short red one is significantly smaller now too. And then I'm going to do the same process for my blue. I've got a short blue chromosome and a long blue chromosome. You can see represented by a dotted line that our nuclear membrane begins to break down. Remember that we learned about a structure found inside the nucleus called the nucleolus, which was a ball-shaped structure that makes the ribosomes. And we didn't illustrate this in the earlier cells, but that also disappears. So primarily the only things inside of this nucleus that is now disappearing is the DNA. In Unit 3, we also learned about a specialized structure called centrioles. Centrioles. Centrioles are a structure that helps organize DNA during this process of mitosis. And it's in this prophase stage that the centrioles begin to separate towards opposite poles of the cell. The centrioles were illustrated using rectangular tube shapes. And this is one set of centrioles which earlier in this process replicated to make another set of centrioles. And that other set of centrioles is starting to migrate towards the other side of the cell. So they originated here from this set, and now they're moving to the opposite side of the cell. What's cool about the centrioles is that another structure is associated with them, and this structure is called spindle fibers. Spindle fibers begin to form, and these are long, protein-rich tubes that grow out of the centrioles, and they will help attach to the chromosomes to organize them later. So essentially this is just the beginning stage of them forming. A memory clue to remember prophase is that the prefix pro is similar to the prefix pri, as in primary, which means first. It is the first phase of mitosis. Now let's dive into the second phase of mitosis. The second phase of mitosis is called metaphase. You can see based on our image that the nucleus has completely disappeared by metaphase. The spindle fibers, which I used orange for, spindle fibers have attached to the centromere of each chromosome and arrange the chromosomes single file down the middle of the cell. So I'm going to start by drawing my centrioles. The centrioles were those tube-shaped structures that are now at completely opposite poles or sides of the cell. And we had 4 chromosomes. They are going to line up single file down the middle of the cell. It doesn't matter what order that you draw them in. I'm just going to start with the long red one. So you can see that these chromosomes are going to line up so that on each side of that middle of the cell there is one chromatid, okay? So keep that in mind. I'm going to erase that though. If you want, you can use that green color again to kind of illustrate that each of these is a sister chromatid, and they are going to be lined up down the center of the cell, represented by my green dotted line. Now I'm just going to draw the short blue one here. I'm going to draw the long blue one. Doesn't have to be perfect. And then the short red one. Remember that if this was an actual picture of your cell, there would be 46 chromosomes lined up down the middle of the cell. And we had described how the spindle fibers had been attached to the centromere of each chromosome. So when you think about what happens in between these two phases, these spindle fibers would have attached to the centromere of each chromosome and pushed and pulled and arranged these chromosomes to line them up. So we are kind of not easily illustrating one of those steps, but I will show that to you in a video that I will link in addition to this one. So we're just going to draw the spindle fibers as having been attached to the centromere of each chromosome. And that, in a nutshell, summarizes the process of metaphase. The spindle fibers attached to the centromeres of each chromosome and arranged those chromosomes so that they lined up perfectly down the middle of the cell, so that one chromatid of each of the 2 chromatids will be pulled apart to opposite sides of the cell in the next phase. The memory clue I use for metaphase is that meta means middle. So M for meta, M for middle. Let's dive into the next phase of mitosis, which is called anaphase. A-N-A, and I was using all caps, A-N-A-P-H-A-S-E, anaphase. You can see based on our picture that the cell membrane is beginning to expand during anaphase. During anaphase, the sister chromatids separate. Sister chromatids separate and are pulled away and apart. So at the end of this phase, each pole or side of the cell, here's one side, here's the other side, each pole of the cell will have a complete set of the original chromosomes. So let's illustrate what this would look like. Again, if we scroll back up, what we're going to see happen here is that the chromosomes are going to split at the centromere, straight down the middle. So this chromatid will go that direction, this chromatid will go that direction. So we're going to illustrate that as if we're in the process of that happening here. So I'm going to start by drawing my centrioles. Okay, I'm going to draw my spindle fibers, and you can see that I'm drawing them shorter because they are already pulling the chromatids away and apart from each other. I think I drew it with my long red chromosome on the top here. And I'm going to draw it as if it's being pulled, like as if you were pulling string along a table, which means that it's going to kind of look in this shape where it's like this instead of just straight. It's like pulling string from one side of the table to another. Actually, I think that deleted it. So instead of drawing it straight, I'm drawing it kind of curved as if it's being pulled. All right. The next chromosome I had was the short blue and then the long blue. So I'm going to draw them in the same order. Short blue, long blue, and then the short red one. You can use arrows to kind of show direction of movement here. So you can see that the chromatids separate to opposite sides of the cell. The memory clue I use for anaphase is that during anaphase, the chromatids, emphasizing, move away and apart during anaphase because they all start with A. So that's one memory clue you can use. The last major phase of mitosis before we get to cytokinesis, the last phase of mitosis is called telophase. Telophase. You can see based on our cell shape that we're starting to see that the cell membrane is pinching inward as the cell is now getting close to being completely divided. The chromosomes begin to uncoil and reform the structure called chromatin, which is that loose and uncoiled shape. So if we were to draw that on each side of the cell, again, if we were to look up here, the cell on the left will have one long one short blue, one long blue, one short red, and the opposite side will have the exact same. So we're going to draw that over here. We're just going to start drawing them loosely uncoiled. So we've got long red one, short red one, long blue, short blue. Same thing on the right side. So the chromosomes begin to uncoil and form chromatin. The spindle fibers begin to break down. So they're no longer there. The centrioles are still there, but the spindle fibers are no longer needed because the DNA has now been organized and separated into the different poles or sides of the cell. You can see that new nuclear membranes are forming. New nuclear membranes form, represented by the dotted lines here. If you need to label that, you can. And the memory clue I use for telophase, telo equals 2 cells begin to form during telophase. If you were to compare what we see here to what we saw at the beginning, remember at the beginning of this process we had one cell that had 1, 2, 3, 4 chromosomes. We now have 2 cells, 1, 2, 3, 4 chromosomes in each. So you can see those 2 distinct cells already during telophase. So the process of cytokinesis is the last major phase, and cytokinesis is where we officially divide the cells. It's the cell cutting phase. So the prefix cyto means cell. So for example, you learned about the cytoplasm, that is the fluid in the cell. So cytokinesis Cytokinesis literally translates to cell cutting. This is where the cytoplasm splits into two, and the cell membrane fully forms between the two cells, forming two individual or separate cells. Again, the memory clue, we've got cyto equals cell, kinesis equals cut. Okay, so if we were to illustrate this, it's essentially the same thing that we drew before. I'm not going to draw the centrioles because they're just not necessarily needed. We've got one long, one short red, long red, one short red, one long blue, one short blue, one long blue, one short blue. If you take a look and compare that to the cell that we started with 4 chromosomes, 2 long of each color and 2 short of each color, scrolling all the way back up, you can see that we now have essentially created 2 cells that were identical to the original cell. Pretty neat if you think about it. So scroll back all the way down. We've got a couple little things to finish up with here. So, mitosis memory clue. How do you remember these phases and the order in which they go? I use prefix. Many people use this prefix to help them remember this. It is called IPMAT. Oh, you can't see that color very well. IPMAT. Interphase, prophase, metaphase, anaphase, telophase. If you wanted, you could add the C at the end, IPMAT-K, but IPMAT will help you remember these phases in order. If we were to compare this process in animal cells versus plant cells, the majority of the process is identical. The part where it differentiates is the end process of cytokinesis. In the picture on the left, you can see that this represents an animal cell where we can see that these 2 cells are pinching inward through what we call a cleavage furrow. So this furrow means that it's dividing, like forming a ridge, and then it fully divides and kind of pinches. In comparison, in the plant cell, what happens is that vesicles of the cell wall start to add up in between the 2 nuclei, and eventually those vesicles form one really long one till eventually it fully divides. And these cells stay pretty much attached to each other, but we have the 2 daughter cells here at the end versus the 2 separated daughter cells here in an animal cell. So it's kind of one small difference between the 2. Let's do a quick summary of what you've learned here about the process of mitosis. So answer the questions based on what you've learned. You might need to look back at your notes to do this. So the process of mitosis begins with 1 cell and produces how many cells? The cells at the end of mitosis are usually genetically, are they identical or are they different? Can you list the 5 stages of mitosis in order? Remember that acronym that we used, and we're focusing on mitosis here. So this means you're not including interphase. Explain the role of centrioles and spindle fibers during mitosis. What job do they have during this process? And last but not least, if a cell accidentally skipped metaphase, how do you predict this would affect the 2 daughter cells and the DNA they each inherit? So answer each of these questions, bring them with you to our next Zoom class, and we will talk about them together. If you have any questions, send me a message. I will also post another link to a helpful video that shows this process overall from as an animation so that it's not just separated images, which we have here. So make sure you check out that supplementary video to kind of fill in the missing pieces in between each of these major distinct phases. Thanks for watching. I hope you have a wonderful rest of your day.